Good afternoon, everyone. I think we have a really great crowd for our Daniel I.C. Wang 2022 lecture. And I'm really excited because we have a wonderful speaker today. But before we get to our speaker, I'd like to give you a little bit of the history of the Daniel I.C. Wang lecture on the frontiers of biotechnology. The person you see is pictured here, Professor Daniel Wang, received his bachelor's degree in 1959 and his master's in 1961 from MIT, and then went on to get his PhD from the University of Pennsylvania in 1963. Now, Professor Wang actually spent 2 years in the U.S. Army at the U.S. Army Biological Labs at Fort Detrick, Maryland, from 1963 to 1965 before he became a faculty member here in 1965. He founded the Biotechnology Process Engineering Center, BPEC, through the NSF Engineering Research Division and acted as its director from 1985 to 1998. And this was really a huge and important center here at MIT because it represented really the foundations of biotechnology and our understandings of biochemical engineering. He was subsequently named as an institute professor in chemical engineering in 2000, and he was given the Temasek Professorship at the National University of Singapore, where he devoted part of his time helping Singapore in their biomedical science research and development. Now, during his illustrious career establishing biochemical engineering, Professor Wang received many awards and recognitions, and these include the ACS Marvin Johnson Award and the David Perlman Memorial Lecturer, the AICHE Food, Pharmaceutical, and Bioengineering Award, Institute Lecturer, and the well-known William H. Walker Award. He was given the Amgen Award in Biochemical Engineering from the Engineering Foundation. He also received the Asia Pacific Biochemical Engineering Award. And Professor Wang has been elected to the American Academy of Arts and Sciences, the National Academy of Engineering, and the American Institute of Medical and Biological Engineering, as well as Academia Sinica in the Chinese Honor Society, and the International Institute of Biotechnology. He has consulted for over 50 companies around the world, and he was on the board of directors and science advisory boards of a number of public and private companies, and while he also authored and co-authored 5 books and over 230 publications and 15 patents. He had a huge impact on the field of biotechnology and biochemical engineering, and one that was founded and seated right here at MIT, so we're incredibly proud of that legacy. I want to give you a few pictures of Professor Wang from the earliest times in his life as a student and in the Army here. And with some of the favorite things he like to do was spend time with his collaborators and his former students. So this is a great picture here with colleagues, including Professor Cooney. Now, this professorship is made possible by Dr. Nubar Afeyan, who is another one of our alum, and in fact is a former student of Professor Wang. Nubar entered biotechnology during its emergence as an academic field and industry and completed his PhD here in biochemical engineering at MIT in 1987. Nubar told me to give the short version, so I'm going to give you a little bit of an abbreviated picture of his career. But he actually became an inventor, entrepreneur, and CEO, and ultimately he launched Flagship. And Flagship has had a huge impact on biotechnology and innovation here in Kendall Square, and includes, as you know, Moderna, which was launched as a flagship company, Kodiak, Denali, and we just could go on and name many, many others. So he has done a huge amount in advancing biochemical engineering and biotechnology around us, and has had a huge impact on human health because of that. He, of course, has had a number of written scientific publications and a huge number of patents, and he's had an affiliation with the Harvard Business School, which allowed him to reach out and engage as a lecturer. And he's also been a lecturer here at MIT Sloan School of Management specifically in the areas of entrepreneurship, innovation, and economic development. And I will just say that he is another amazing part of his history is that he is an immigrant and has been recognized by receiving a number of awards and from that perspective as well. And I will stop there because I saw the look and move to our speaker. Now, Dr. Georgiou is an elected member of the National Academy of Engineering, the National Academy of Medicine, and the National Academy of Inventors, as well as the American Academy of Arts and Sciences. And back in 2013, he was selected as one of the top 20 translational researchers by Nature Biotechnology. He founded GGMJD in 1999, which was ultimately acquired by Maxigen in 2000, Aglia Biotherapeutics in 2013, and Ikena Oncology, and served on the board of directors of both Aglia and Incna. Dr. Georgiou's research is based on the molecular level understanding of human adaptive immunity in infectious diseases and in autoimmunity, and the discovery and preclinical clinical development of protein therapeutics. And finally, the biology of receptors and the engineering of therapeutic antibodies with improved effector function. Notably, he is co-inventor of 5 protein therapeutics that are FDA approved and or in clinical late-stage preclinical evaluation. And we hope to hear a little bit more about how these translations were able to take place. He has also been recognized by many awards, including, like Professor Wang, the William H. Walker Award for Excellence in Chemical Engineering, and the AICHE 2019 Billy and Claude Hocutt Distinguished Chemical Engineering Research Award at UT Austin, and the 2019 Startup of the Year Award. So we're really excited to hear from George, and I'm going to turn things over to him. Come on, George.
Thank you. Oops. Okay. Something is working. Okay. There will be a seminar. First of all, I have to say I'm truly honored and actually emotional to be here, to be giving this lecture for a number of reasons. First of all, because I knew Danny very well. And of course, when I started my career, he was one of the giants in the field. He continued to be. And he was a bigger than life person. And there were 2 things that I found out later you should never do with Danny. One is go drinking, and the other one is play poker. And I did both, and they shortened my life. The first one, because I drank too much, and I have the Greek alcohol dehydrogenase gene, which is not a very good one. The second one, because my wife found out about the poker game. But he was definitely an influence for my generation and for the generations to come. I'm also it's an emotional moment, first of all, because I wasn't expecting that many people here. And also, it's my 5th seminar at MIT. I don't know why, but I've been invited back several times. So now I have an additional responsibility to do something to make sure that your time here is worthwhile. And the only thing I can do is entertain you. And because we're all geeks, I have to talk about science because we find science entertaining. the rest of the world doesn't necessarily feel that way. Parenthetically, if you told me when I was starting out or when I was a graduate student, I would have given a named lectureship at MIT and I would have given 5 lectureships at MIT, I would ask you 2 questions. What have you been smoking? And can I have some? But life takes various different turns. And my life took different turns. Most of them, I wish I could say it was by design because I knew what on earth I wanted to do, but it was all happenstance and being at the right place at the right time, and on occasion not making terribly stupid mistakes. So in any event, I started at the University of Texas my career thinking that I'm going to stay there for 2 years. And here I am 37 years later for various reasons. I have to say that Austin has been a fun place to be. It's not like the rest of Texas in a good way. Anyway, and it's been good for my career. For many years, I was working with bacteria. We developed a number of technologies because, well, I was a molecular biologist at heart, but I was in a chemical engineering department, so I had to do something useful. So we developed a number of technologies. We licensed them to several companies. There were interesting stories to tell about some of these technologies that I don't have time to repeat. A major company licensed one of our technologies, and then 2 years later, they wanted to license it again. And I gave them the wrong answer. I said, but you already have it. I could have said, yeah, sure. Yeah, we'll take your money. And one of these has been actually quite useful in terms of making a number of small protein therapeutics like parathyroid hormone and Lucentis, which is an antibody fragment, and so on. But around 2008, I decided that I wanted to make a transition, and I moved into therapeutics and human biology. We already knew how to make proteins and engineer proteins. And also something very important happened in my life. I had a really bad skiing accident because unlike Professor Cooney, I love skiing, but I'm not a very good skier. So I broke my leg and I was in bed for about 2.5 months. And then I reread immunology and I thought, oh, gee, that's what I want to do. And this is a true story. And now, why did I spend 2 months reading immunology? Well, if you're a geek and you have to stay in bed, and I had to stay in bed because they couldn't put a cast on my leg, then you get bored and you cannot watch TV for a long time or read books. But immunology, it was fascinating. So we turned to immunology and therapeutics. And since 2008, we've been working on understanding the circulating antibody repertoire, the antibodies that we have in our blood and how they impact disease at a molecular level. And I'll explain in a minute what that is. And also on therapeutics for enzymes and as well as second-generation therapeutic antibodies. And for a number of reasons, this all worked out reasonably well. And we ended up, as Paula said earlier, with a number of therapeutics that have either been approved, well, one of them has been approved, one that is about to be approved in Europe and hopefully eventually in the US, and several in clinical trials or that are about to hit clinical trials. We were, as a lab, and this is what I enjoy doing because it utilizes some of my engineering skills, we're interested in both the discovery process, like how to make something, understand where to use it and why this particular molecule may be useful in a way that other modalities are not feasible. And also do some of the early, like blue-collar work, the development work, develop a therapeutic candidate, look at the first stages in developing a fermentation process, production, and so on. So anyway, it was a useful and interesting experience. And I owe it to Bob Langer when I founded Aglia. And by the way, founded these companies and then went out and raised money for those companies, which was not easy because I had to deal with investors and I didn't know how to deal with investors, but you learn things. He said, you have to be on the board of a company that you found. And it was invaluable experience. I wish I could pay him back. It cost me, again, it took some toll on my life because I was not a professional entrepreneur. I didn't know what I was doing. But nonetheless, that's how you learn, right? You get into a situation and you learn. So Eglia, commercially, is still publicly traded. I was on the board when it was private, and I'm still on the board, and then when it was public for many years. Commercially, it wasn't a very successful company because of management issues and other issues, but it did get one drug through the finish line, and hopefully we'll get a second drug through the finish line. Anyway, I'm not going to talk about these programs. They're quite interesting on their own right. These are programs for orphan diseases, genetic diseases, and also in one instance for cancer, although the company is not pursuing the cancer angle, and I'm planning to pursue that separately. We continue along those lines with different, mostly enzyme therapeutics, because we think there are needs that can be met. But instead, I will talk about something that's more accessible to a general audience and probably more closely aligned to what people think about these days, which is infectious diseases and antibodies. So, and what I'll discuss is the circulating antibody repertoire. In other words, when you have COVID or when you have tested, you have gotten the vaccine and somebody tells you, oh, you have a titer of antibodies to COVID. What does that mean? How many different antibodies do you have? How long do they stay around? And what do they do? Which one of these are helpful and which one of these may not be helpful? And what else is it that we don't know about the circulating antibody repertoire? But before we talk about antibodies in circulation, it's important to provide some background. So humans have 5 different kinds of antibodies. They all have the same general structure. This is a sophisticated audience in biotechnology, so you all know that antibodies are Y-shaped molecules. And they comprise of 2 parts, one which is the binding part and one which is the part that is recognized by other immune cells and enables the immune system to clear a pathogen. The most relevant part or relevant class of antibodies is the IgG antibodies, which come in 4 flavors. And they are the principal means by which we clear pathogens. Both in circulation in the body as well as in epithelial tissues, in the lungs, in the GI tract, and so on. Then there is IgA. IgA comes in 2 shapes, monomeric and dimeric. Dimeric is all exclusively or nearly exclusively found in epithelial tissues, and monomeric is found in serum. We don't know exactly what the monomeric IgA does in the body. In fact, it may have both a role in clearing infections, but also it may have an anti-inflammatory role in some instances, and that's actually quite interesting. Then there is IgM, very large antibody molecules that form the first line of defense. They can basically recognize generically, in a manner that we don't really understand, pathogens or things that are not our molecules, and clear them. And also they have another very important function. They clear parts of dead cells. They help clear parts of dead cells which could otherwise accumulate in the body. Then there is the obnoxious IgE, which probably evolved because of the requirement for having a mechanism to destroy parasites. We no longer live in a parasite-filled world, at least not in this country, which is perhaps part of the reason why IgE now is very much associated with allergies and pathogenic events. And then there is IgD that nobody cares about. Okay, so in the blood, most of the circulating antibodies are in the form of IgG. There is some IgM and some IgA. All of it is monomeric IgA, not the dimeric form, which is secreted. On the other hand, in the respiratory tract, we have a very large amount of IgA with smaller amounts of IgG and very small amounts of IgM. Well, it turns out that, for instance, for COVID, most of the protection, perhaps, even in the respiratory tract, comes from IgG, although it's not exactly certain that this is the case and that the IgA does not contribute. These are details. First, let me tell you a little bit about B cells and antibodies and sort of give you an overview of how antibodies are made. We have about 180 billion B cells, and the B cells are the cells that make antibodies in our body. And if you ask me how I know that, the answer is I do not, but I trust the people that did the calculation, and these are Rob Phillips at Caltech and Ron Milo. They've written actually a superb book on quantitative biology, which is pretty simple but amazing in terms of giving you a perspective. It's the chemical engineering. It's a book that was written by applied physicists, but should have been written by chemical engineers. And from other sources, we know that most of our B cells like to hang around in our lymph nodes. Humans have about 600 plus lymph nodes, which is part of the reason why researchers on occasion say, we'll take a lymph node or two away for research purposes. You don't need that many. I don't think that's a good idea, but it does happen a lot. There is a large number of B cells in the spleen, bone marrow, and of course in the GI tract. So all B cells begin their life in the bone marrow. They are hematopoietic. They are derived from hematopoietic progenitors as naive B cells. Naive B cells means they have very low affinity for antigens. The diversity of the naive B cell repertoire is approximately 10 to the 7, 10 to the 8. We don't know exactly what it is, but there are about roughly 30 to 50 billion naive B cells in the human body, mostly circulating throughout our body. And at some point, they encounter, say, a foreign molecule, say a SARS-CoV-2 protein, or some other protein that we know is not ours because there is inflammation at a particular site, and therefore there is the right milieu, the right environment for B cells to be activated and now begin to bind with very low affinity to that protein. Well, they will undergo a process of mutagenesis, which will result not only in the increase of the affinity of the antibody for the target molecule, but also a number of changes in the B cell physiology itself. These B cells will become B memory cells, and the B memory cells are what contributes to long-term memory, the immunological memory that we all have. For those of us who are blessed to be over 50 years old, we were vaccinated with the smallpox vaccine, the old smallpox vaccine, and we have the mark of the smallpox vaccine, which means, given the epidemiological data, we're all protected from monkeypox because the old vaccine protects you against all kinds of viruses of this family. Anyway, the reason why we're protected is to a large extent because we have B memory cells. Also, we have a small amount of antibodies circulating that are still giving us protection against things like these kind of viruses. Anyway, we have memory B cells. The memory B cell diversity is actually very large. There are very interesting cells in many ways. First of all, they have lengthened telomeres. Normally, as you get older, a cell gets older, the ends of its chromosomes shrink, but in B cells, and that was realized recently, they actually get a little longer. We don't know why. And also there are cells that are heavily mutated, and they're heavily mutated because in order to make very high affinity antibodies, there is a mechanism that is responsible for mutating the antibody sequence, and the antibody sequence is mutated at a frequency that we now know is about 300,000-fold higher than any other part of the genome. But the genome is very large, so these B cells are hit with mutations. And this is part of the reason why leukemias are the most common form of cancer, or B cell leukemias. A small number of memory B cells will mature further into antibody-producing cells. And the antibody-producing cells eventually, or some of these, will go into your bone marrow and will find residence in the bone marrow, and this can be in the bone marrow and have a half-life between 20 and 40 years, maybe longer. They produce antibodies at a rate that's approximately 4 or 5 times higher than the best CHO cell processes that Genentech has. They are super oxidatively stressed because they produce proteins that have a lot of disulfide bonds, and yet they survive for a long time, unless in some cases they go bad and then they cause multiple myeloma, which can happen later in life. But the plasma cells, these cells that are found in, I have a pointer somewhere. Nope. Well, okay, these cells, we know there are about probably 5 billion cells in the bone marrow, and they produce the circulating antibodies that confer protection to everything that we're exposed to on a regular basis. That means they produce IgG. IgG antibodies have a long half-life, and they stay in circulation for about a month. We don't know what happens when antibodies age, but eventually they will be eliminated. In other words, as they age, an antibody is produced. It's now a young antibody, but it will be subjected to a lot of abuse in your bloodstream through oxidation and other processes. Things will happen, but eventually it will be degraded because it has to be replaced. But they're produced continuously. In the serum, you have between 6 and 17 milligrams per mil of antibodies. And this is clinically defined. If you go to your doctor and you have less, they will call you. They will tell you that you have an immune deficiency. You may have no symptoms, but you have an immune deficiency. If you have more, you have a hyperglobulinemia. You may have no symptoms, but you have some disease that may end up being a problem later on in life. We know from our measurements and from other calculations that we have approximately 20 to 15,000 to 20,000 different families of antibodies circulating in our blood. And these 15 to 20,000 antibodies are what protects us from a large number of viruses and pathogenic cells, which to me is pretty amazing. I mean, I don't know how that works if you think about what we're exposed to, but it seems to work just fine. Now, I have to point out that, and this is a chemical engineering audience, these are antibodies that are present in sufficient concentrations to be biologically relevant. What that means is that they are present at concentrations that are comparable to the equilibrium constant for binding pathogens. You have antibodies that are present in what we technically call nanosquat amounts. Well, those are not meaningful because they cannot bind. There's not enough antibody to bind and be physiologically relevant. So all we know about B-cell immunity, and that's pretty amazing because there have been 5 Nobel Prizes on antibodies so far, the most of any other field, by the way, in the biological sciences. All we know about antibodies and antibody generation has come from bulk assays. We measure the mean of a population. So Dr. Afian is here with us today. If we take our mean net wealth is actually quite high. But that doesn't mean that necessarily it's an average of a population. So again, averages tell you only one thing. They don't tell you the full story. And it's pretty remarkable. And when I was trying to think about this problem, while lying in bed, waiting for my leg to be fixed, I asked my immunologist friends, how many antibodies do we have in our blood? And the answers varied quite a bit, between 2 million, I don't know why they came up with that number, to why the hell should I know? Or why are you asking? So it turns out that actually identifying the antibodies in serum is not an easy bioanalytical problem because antibodies are all very similar. They vary widely in the range of concentrations, and they're produced by cells that are not accessible. If somebody could drain your bone marrow and isolate those cells that produce antibodies, then they could presumably tell you what antibodies you're making, but you need your bone marrow, so I don't think you would want to participate in such an experiment. So there has to be another way to do this. So we set out to solve that problem, first by developing methods to analyze the B cells that produce antibodies. And Dr. Dekowski was really seminal in these studies. Yes, that guy over there was really seminal in developing some of these methodologies, which proved to be very useful for analyzing very large numbers of B cells very quickly. And then the other part of how we go about analyzing the antibodies in circulation is using very high-resolution LC-MS technologies and running very long runs so that we can identify peptides, we can identify portions of antibodies that are signifiers of a particular antibody, and then we can quantify them based on the liquid chromatography component of LC-MS. So what do we do with these experiments? Well, we do what is technically called CDRH3 spectrotyping. We analyze and determine the most hypervariable part of the antibody. We quantify from the liquid chromatography peak intensity. The sensitivity is very high. It's way below nanomolar. The coverage, the number of antibodies that we can detect, the percentage of the circulating antibodies is also very high if you do the experiment right, at least 80% for most normal-looking antibodies. And then once we have the identifier of the antibody, then we can go back to the sequences that we derived from the B cells that are unique. It's the directory. It's the phone book of your antibody system that we derive from the B cells, and that's what Dr. Dekowski built as part of his PhD dissertation. We can go back and determine the full sequence, produce the antibody, and characterize it. Okay, so that's what we do. There's a lot of papers that describe this, but we stopped making platform technologies because after a while you say, okay, we develop technologies, now let's do some biology and not switch to something that is more satisfying. Well, not that platform technologies are not satisfying, but it gives you some insights on human biology. So what do we know about the serological antibody repertoire? First of all, in every instance that we've looked at, and now we've looked at more than 300 people with various diseases, autoimmunity, after vaccination, probably more than 500. In every case, the antibody repertoire, what gives you a titer in an assay is really restricted. You only have about roughly 30 to 50 antibodies. And that is either protective against COVID or you're out of luck. Always, the antibody repertoire is polarized. There are 2 or 3 antibodies that are present at very high concentrations and dominate the response. So ideally, you would like to have a vaccine or an infection that primes these antibodies that are super important for protection. Some rare antibodies can also be relevant, and also maybe the aggregate of antibodies may be relevant to at least helping you be protected against disease, simply because they coat the virus or the pathogen and they allow it to be engulfed by cells of the immune system. But ideally, you would like to have antibodies that block the virus or they block the pathogen from causing infection. Now, many of the serum antibodies that we found, there are many examples that we published and some that I will discuss later on, provide unanticipated insights on how you can be protected against infection, or in the reverse, how antibodies could contribute to autoimmunity. There is a low degree of overlap between IgG and IgA antibodies. It's actually quite interesting. I don't have enough time to talk about the implication of that in disease, and it's something that we're exploring right now. 2 things that I find very fascinating, but the work is not completed. A lot of the antibodies that we have in circulation do not just bind what they're supposed to bind, but they exhibit a property called polyspecificity. In other words, they're somewhat sticky. This is important. All the therapeutic antibodies, to my knowledge, that we have, and we have about 100 therapeutic antibodies approved by FDA, have been selected so that they're highly selective to bind only to their target. Herceptin binds only to HER2. It doesn't have binding to other molecules. But many of the antibodies and many of the very good highly neutralizing antibodies in influenza and HIV are a little sticky, and we don't know why that stickiness is selected and why it is there. We have some interesting hypotheses, but they're hypotheses right now. The other thing that I care about, because I did the analysis on myself, and also most of us that are over 50 probably should care about, is that everyone who's over 50 has antibodies that are present at insanely high concentrations. Now, this is not clinically detectable, but when I say insanely high concentrations, I mean concentrations over 100 micrograms per mil, and sometimes over 500 micrograms per mil, or even a milligram per mil. That's a high concentration, especially for IgA antibodies. I measure my caloric consumption in McVitie's chocolate-covered cookies because I'm addicted, and if I don't have 3 cookies, actually I travel with McVitie's chocolate-covered cookies with me because I have to have 3 cookies with my first coffee in the morning. So I estimate I have to eat 2 cookies a day just to replace the IgA antibodies that are at large levels, and then they dissipate. I basically piss them off because that's how you replace antibodies. They are eliminated in the urine during the day. But anyway, why do we have these highly expanded antibodies, and what do they do? Are they just abnormal because the immune system works that way, or are they precursor, as I think it is, of pathologic conditions that may predispose to either multiple myeloma, a form of cancer, or some form of immune dysfunction that we can discuss later if you like. Let me give you a few examples of what these techniques have allowed us to learn so far. And the first one is related to COVID because everybody cares about COVID. I care about COVID. I had bad COVID about 3 months ago because I was trying to be very smart. I work on COVID. I thought, okay, I had my 3rd shot. In August, I will wait for Moderna to come up with a booster that's a better booster. And I held off. I didn't get the 4th shot. And 9 months later, I got COVID, and it kicked my derriere. So now I know better. After I learned that, by the way, several people in my cohort of scientists that are working on COVID, they were also holding off, and as soon as they saw, oh, yeah, he got bad COVID, maybe we should get the 4th shot and not wait for the booster. So now, the reason why, and this is, by the way, we published a paper in Science, and it caught the attention of the NIH director who blogged about it, and this is from his blog, so very honored, so I had to show you this and advertise our work. And basically, antibodies are elicited against the spike protein, the major infectivity protein, the S protein or spike protein in COVID. And if you're lucky, these antibodies will block the protein from binding to the receptor, the ACE2 receptor that now everybody knows is the target for the COVID virus for internalizing to the cell. There are 3 major domains in the spike protein of interest. The RBD domain, the receptor binding domain, is the target protein, the target component that is targeted by all anti-COVID monoclonal antibodies that are used therapeutically. The N-terminal domain, which is essentially a linker domain that has a high degree of variability. The entire protein has a high degree of variability. And the S2 domain, which is actually not subject to a lot of mutation. It's rather, unlike the other parts of the virus, it's quite conserved for a number of reasons. Antibodies to S2, with very, very few exceptions, do not block the virus from infection, although they could be assisting with other ways of clearing the virus that perhaps are less important. So what did we do? We took serum from infected, naive patients, in other words, patients that had not been vaccinated. That was early in the pandemic when nobody was vaccinated. And we analyzed the serological repertoire, and this is an example of the serological repertoire of one of our donors. And it turns out, as you can see here, each of these bars represents the relative abundance of an antibody, of different antibodies. And you can see we monitor whether these antibodies bind to the RBD domain or some other portion of the virus. And most of these antibodies actually do not bind to the RBD domain, the domain that's the most interesting and important target for neutralization, but they bind to other parts of the virus. And in fact, only about, on the average, 10 to 15% of the antibodies elicited by primary infection are directed against the RBD. This is an example of one donor whose entire repertoire of antibodies, the entire titer, and we actually verified that, was due to 6 antibodies. So there are 6 antibodies. One of them should be not neutralizing. If it's not neutralizing, you have a problem. Well, it turns out only one of these antibodies bind to the RBD domain. That antibody was not neutralizing. However, there were 2 antibodies that bind to the NTD domain. And these antibodies are protective in mice. These are mice challenged by a mouse-adapted strain of SARS coronavirus. And you can see that in one particular case, this particular antibody enabled the mice to basically clear the virus rapidly. All the other antibodies did not. So a single antibody, and we know from a number of studies that I'll discuss in a minute, that was directed to the NTD domain, the N-terminal domain, was responsible for the neutralization activity of this of the virus in this donor. So you have a polyclonal response, but it's only one antibody that really does all the hard work. Well, it turns out, and this is actually, I think, relevant, perhaps to the broader population, it turns out that this antibody is quite interesting and unique, and it's encountered in many donors, and it's very easy to elicit because it doesn't require the immune system to do a lot of work. It's not somatically hypermutated to any great degree. It's essentially nearly germline. So variants of this antibody are found in multiple donors. It's what we call a public antibody. It's actually quite interesting because this particular VGene family is rather unique. So in collaboration with Jason McClellan, who actually is a colleague of mine, and he's the one who invented the stabilized form of the coronavirus, SARS coronavirus protein, the spike protein, that's been used in vaccines. We saw the crystal structure, where they saw this crystal structure, and we know exactly how this antibody binds. To make a long story short, the message here is that basically, for some people, antibodies that are pre-existing are actually able to confer a level of protection because it doesn't need much for this particular antibody to be able to confer protection. Now, an interesting question for those of us who are vaccinated is what happens when you get vaccinated. It turns out what happens when you get vaccinated, you get a very broad response, an expanded repertoire of antibodies that are mostly directed to the receptor binding domain. And that's a good thing because these antibodies are more likely to be protective against very different variants, which unlike NTD antibodies, which may or may not be protective because the variants change their NTD domain quite a bit. So in this slide, each of these antibodies, each of these lines against a different antibody, these are high responders. These are responders with the Pfizer vaccine, but we see the same thing with the Moderna vaccines. The vast majority of the antibodies in the serum are antibodies to the RBD, with a few to NTD, and very few to the S2 domain. But what happens when you get a breakthrough infection? And that's interesting. When you get well, it's interesting to me at least. When you get a breakthrough infection, okay, what happens is the antibodies to the S2 part of the protein, which is not neutralizing, are boosted preferentially to those that are binding to the RBD. Now, you get infected, your immune system is working overtime, it's going to make higher amounts of antibodies, of all antibodies, including the RBD antibodies, but it becomes polarized to start making antibodies to the part of the virus that, first of all, more constant and not as subject to mutation, and therefore, and secondly, not as neutralizing. And we have one donor that was infected twice, and you see even more enhancement of those antibodies. So don't get infected with COVID too many times, or if you do, get some booster again to diversify your response, because otherwise it seems that your response will begin to be increasingly polarized towards antibodies to the S2 domain. This does happen very often with repeated immunization, and this is part of what we see in influenza as well, and part of the reason why the influenza vaccines don't work very well, because we keep making antibodies that are basically targeting parts of the virus that are not the virus that is infecting us in this particular year. Speaking of influenza, so influenza is actually another infectious disease. The influenza season is upon us. Hooray. Now we have to worry about COVID and influenza. And by the way, there is a remarkable number of co-infections. So I wear a mask when I travel. I highly recommend wearing a mask when you travel, probably when you're very crowded spaces, but everybody here is presumably vaccinated. If I was in Texas, I would definitely be wearing a mask. So now, influenza is an interesting virus. Well, they're all interesting in their different ways. But the influenza virus basically comprises of different families of viruses, the A and the B strains. The A strains come in two forms. The A strains, by the way, are the strains that circulate between animals, like birds and pigs and other animals, and bats. Those darn bats are everywhere. And the B strains. The B strains circulate only in humans and in seals. I don't know why seals, but that's established. Now, what has been causing the latest pandemics and most of the deaths has been the group 2 influenza strains, primarily H3s in recent years. And this is a chronological order of the different pandemics from the last few years. In 2009, we had the sort of swine flu. Fortunately, we were able to develop a vaccine very rapidly, good old-fashioned egg-based vaccine, but it worked. It stopped the pandemic in its tracks. But ever since, most of the influenza outbreaks and deaths have been primarily because of H3 strains. So all our assays for developing antibodies or for developing vaccines, or for that matter, for therapeutic antibodies, are based on whether an antibody can block a virus from infecting cells. And this is the clinical assay. The clinical assay actually that's used by the FDA is a very easy assay to perform, but it's a dumb assay. I mean, it's a dumb assay not because these people are not smart, but because implementing very good assays is costly and not clinically as feasible because we don't have the infrastructure to do so. But we rely on the production, on whether we produce neutralizing antibodies to the virus. Now, the more neutralizing antibodies to the virus you have, the better protected you are. However, the more neutralizing antibodies to the virus you also have more antibodies, period. So you don't know exactly which is protecting. So this is an example of a mechanism of protection against influenza that doesn't rely at all on neutralizing antibodies. So when we started working in influenza, and that was a few years ago, we realized that there are some antibodies that are quite prevalent in the serum of some of our donors that bind to all known strains or most of the known strains of influenza, which was very odd because there is a high degree of sequence diversity, so that this is rather rare. And when we looked at these antibodies, we said, oh, well, the paper actually was interesting. The paper was half written, and we said, oh, yeah, we make a lot of antibodies that don't do anything because they bind to some part of the virus that the virus doesn't care about. And a very bright and exceptional researcher. Florian Kramer was in the audience when I gave this talk and said, wait a minute, let us take these antibodies in mice just to see what they do. Maybe they'll do something interesting. And when we tested these antibodies in mice, we saw, in fact, these antibodies are actually protective. They don't block the virus, but they protect from death the animals, and they actually reduce viral loads in lungs. And the reason why they do that is because they bind to a portion of the virus in such a way that they can engage immune cells to clear the virus by eating up, first of all, the infected cells, and secondly, the viral particles themselves. Well, we found that, and we published it actually in collaboration with the NIH. We saw the structure of that antibody. These antibodies have very high binding breadth. They're found in high concentrations in the serum, at least in our donors. And then after that, they were found by many others, and we were actually a little upset because our first paper was sent to Cell. Cell did not take it out for review, but then they took out the second paper and the third paper, and they were published in Cell. But anyway, can't complain because the paper was published in Nature Medicine, so I whine because I like to whine because that's part of the job of being a professor. So in any event, these are antibodies that are quite common in influenza. They are not protective. Your epidemiological assays as to whether a vaccine works or not will not pick them up. But there are mechanisms of protection that we don't know as much about and we cannot account for very well. Now, I mentioned something earlier about how that happens when you revaccinate and revaccinate and revaccinate. This is a study that we did with Antonio Lanzavecchia, a very noted virologist, and well, actually immunologist, but has done a lot of work in immune responses to viruses, where we took a donor. We don't know the identity of the donor, other than it's a famous Italian engineer, age 58. Well, because Italians are very good at talking, and so we know that, anyway, this particular person was vaccinated with the same vaccine many, many times, and we asked over a period of many years, which is what happened to us because we get vaccinated with influenza for many years. And it turns out, when you get vaccinated, what you do is you're making more and more and more of the same antibodies. You don't diversify your antibodies, and this is a problem with getting too many vaccines of the same kind. You make certain vaccines become entrenched. This problem or this property is called original antigenic sin. It's not a sin, but it's a nice name. Or more properly, antibody imprinting. Your immune system is imprinted. When you actually are first vaccinated or infected with influenza, it imprints you, and then that imprinting has lasting effects on your immunity for many, many years to come, which probably is the reason, speculation, but actually not completely unfounded, part of the reason that a few of us somehow never get flu, and they brag about it and make everybody else feel bad, but something happened and their first response was protective and continued to be boosted over the years. Okay, I talked about the IgA repertoire, and IgA antibodies can be made either by starting with IgM, which is the prototypical form of antibody in naive B cells, or by crossing from IgGs. We asked, what is the relationship between the IgG and the IgA population in humans, especially after vaccination? It turns out that the IgG repertoire, the binding specificity as well as the sequences in the IgG repertoire are quite different. We don't know why they're different, but they're quite different. After vaccination, you boost the antibodies that are similar in sequence, IgG and IgA, but these decay over time, and then you go back to the basal level of roughly between 8 and 15% overlap between the repertoire. By the way, the similarity between the IgA responses and the IgG responses varies with age, and we don't know why either. There's nothing unique about your IgA and IgG antibodies. They all look the same to a first approximation. They have the same number of mutations, they have the same features in terms of gene usage, everything seems the same, but you make different antibodies. And I don't know why, I don't think anybody knows why you make different IgA antibodies versus IgG antibodies, but that's actually quite important in terms of the antibodies that are going to be able to transverse, to cross from the serum and go to your epithelial surfaces and eventually protect you from disease. So this is coming up. We don't know all the details yet. Finally, to keep in time, a few things about polio. We have this polio work that's been languishing forever, and now it's become popular, so now we're revisiting, and we're finally going to write the paper. But this is like work that was in the cell for about 5 years. Brandon is laughing because he was a graduate student, and we kept saying, oh, yeah, we're going to publish this paper one day. But now polio is back, so it's more urgent. So we looked at a number of people that were mostly in the 60s and 70s and had been vaccinated against polio when they were children. And they were reboosted with an inactivated polio vaccine. Well, it turns out that the polio vaccine is super effective. So a very small dilution of the antibodies that blocks the virus from infection can be protective. So we looked at these people that were vaccinated between 50 and 70 years earlier and asked what happened to the serum. How do they still have antibodies against polio? And by the way, in order to do this experiment, we needed polio vaccine and a lot of it to be able to do affinity chromatography with these antibodies. So we got about 200,000 doses of the polio vaccine. It was a technical lot that was rejected by Sanofi, but it was good enough for us. So they were kind enough to give it to us. So it turns out that even after all these years, people had protective serum antibodies patrolling their circulation and their tissues to clear polio. When they were essentially challenged with a vaccine, you see a huge increase in the level of antibodies that they produce and in the level of neutralizing antibodies. So this is an example of a 74-year-old participant. And as you can see, upon vaccination, some antibodies are now that were already existing in the serum but are at low amounts, now are boosted by insane amounts, and they are super protective. And it seems that the protective antibodies are boosted preferentially. This is true for all the donors. And what's also very interesting and complex, and I really don't want to go into details, these people have circulating B memory cells that have been circulating for, again, 60 to 70 years that are specific to antigen. And as soon as they see poliovirus, they start proliferating and they start expanding and then producing even more antibody to clear the infection. So polio is an example of a spectacularly good vaccine. The smallpox vaccine that I got is another example of a spectacular good vaccine. We know that the longevity of the response of protection is about 60 years. I mean, it's probably longer, but we don't have that many people that have been followed. We don't have that many 100 years or whatever. Kind of do the math now, since the smallpox vaccine became available and widely disseminated. And by the way, an interesting, probably many of you know about this, an interesting caveat, we have about 200 million doses of the smallpox vaccine that we got vaccinated with as a strategic reserve, but we cannot use it because when you get a smallpox vaccine, there is 1 in a million probability that you can die and maybe a 5 in a million probability you can get serious disease. Remember the backlash against COVID vaccines? Imagine what would happen if the government told people, hey, you can be vaccinated with this old vaccine. There's a newer vaccine that's safer, but we don't have it. But in the meantime, we have this other vaccine that you could be vaccinated, but somebody could get sick. So it's never going to be used, but at least there is a stockpile in case there was a real serious emergency. And that actually, and I wanted to close with this because that points to the complexity of social as well as biological issues that underscore both vaccine development and dissemination, and we're all experiencing those. And I live in Texas, where these are more intense. Okay, I'd like to stop here and acknowledge the team. First of all, we have a multidisciplinary team. We're toying away with like different models of being a professor and having an academic groups, and we're trying to basically have a multi-PI group, which we don't know exactly how to work with that. But anyway, we operate as a team. The enzyme therapeutics work that I described before is in collaboration with Dr. Everett Stone. He's a research professor. Dr. Ippolito and Dr. Lavender are also research professors in the immune repertoire field. I mentioned Ji-Wan Lee first, who's now at Dartmouth, only because I talked a lot about the serological repertoire. But of course, all that work was predicated on the brilliant insights of Professor Dukowski. Thank you, Brandon. A number of other very talented people. As you notice, a lot of them are in the Bay in Boston these days. And the current number of students in the field, at some point I thought, maybe, should I be taking more students? Maybe I should do something else in my life. And then I realized, nah, I like what I'm doing. So we took another 5 students last year. And also a network of really great collaborators. And I want first and foremost Dr. Ralph Berrick. I don't know if anybody has heard of Dr. Berrick. He's actually the coronavirus expert. He's a great man. He used to run a very small lab because he was working on this deadbeat virus that nobody cared about. And all of a sudden, he became a celebrity, so much so, he needed to be under basically FBI protection because he was getting all basically threats because especially in the beginning of the pandemic, there were all the crazies that thought that, he engineered the virus together with the Chinese. And it was pretty serious. But anyway, he's a great scientist and a great collaborator. And many others, Steve Harrison at Harvard Medical School, of course, the VRC at NIH. By the way, for those of you who have heard the annoying comments about Fauci and how he was not really a great leader, they should all remember that it was because of the Vaccine Research Center that he founded, and he pushed for many years, and the incredible people there that we have the vaccines that we have right now. So we owe him a lot. I tried to write a letter to the New York Times to say that it didn't go very far. Anyway, because they wrote some opinion piece saying, Fauci kept giving, like, they wanted to appear to be more kind of balanced, and they had this right-wing, obviously my politics are not on that side, right-wing person write a letter basically trusting Fauci, and some of us think that Fauci is an amazing public hero. Thank you very much.
Thank you so much, George. That was an incredible talk, and I think a really relevant one for us today. And I think we all learned something new. Before we turn to questions, I'm going to reach down and pull out a symbol of appreciation for giving the Daniel I.C. Wang Frontiers of Biotechnology lecture. Thank you so much, George.
Thank you. The pleasure is all mine.
Thank you.
And I'm truly honored.
Thank you. And we'll help you get that home.
Thank you.
And in the meantime, we're going to open up to questions, and we have microphones at the ready we're going to be handing out to those who have questions. So here we have a question right now. And then over here, okay. Start here.
Very great talk. I have a question. Like in your slides, you mentioned the antibodies crystal structure, and in this summer, AlphaFold by DeepMind are reported to accurately predict like a 200 million protein structure. How do you think that AlphaFold 2 would help the development of antibody, human health and antibody?
I couldn't understand because of the match. Sorry, the AlphaFold. You say something about AlphaFold or?
How do you think the importance of AlphaFold 2 by DeepMind would to the development of human health and antibody?
Bioinformatics?
Huh?
No. The AlphaFold.
How is useful?
I let Dr. Whittrop respond. Antibodies are very hard. I mean, look, okay, there's so many people working on that problem. I know there's going to be progress, but it's going to be slow because there's a lot of dynamics. And of course, the loops in antibodies that matter the most are very difficult to model yet. So we'll see what happens. And keep in mind, there's another very important issue with conformational dynamics that we really don't think about, but that's well known in the antibody field. Some antibodies can assume more than one conformation, and they can bind to two antigens, and this is part of the way how autoimmunity arises. Whether computational tools in the near future will be able to give us great insights and rapid progress in this field, I do not know, okay? Because of the nuances behind antibodies. Will they be useful? Absolutely. They are already useful in many ways in accelerating the discovery of antibodies, but whether you can basically plug in the sequence of a protein or the structure and come up with an antibody that will bind where you want it to bind, that is probably going to take some time. Dane? Okay, he agrees.
Thanks for a great talk. So 2 quick questions. One, in the context of the aging immune system, the excess antibodies that are produced, especially in the context of MGUS, would you speculate that that is from a single clone of long-lived plasma cell or a general attribute of the aging cells? And then the second is broad, just what is missing from the landscape of platform technologies for pairing B cells with their antigens?
For pairing B, well, first of all, the first question, these are most likely aberrant B cell expansions. They're not necessarily cancerous, but I don't know the reasons why they arise, but clearly there's something there in the way these plasma cells work. There is a formal possibility that they're there because they're protecting us from some obnoxious virus like CMV that is trying to kill us all our lives. But I don't think that's the answer, and I don't have evidence yet that these are, say, CMV-specific antibodies, although it could be another virus that I don't know about and we don't know about. So I don't know, but I think it's mostly a consequence of B-cell biology and the fact that these cells at some point may expand in a benign fashion or, in some cases, not benign fashion. Now, in terms of predicting antigen specificity, I don't know. I wish I could give you an answer, but I don't know how we can predict antigen specificity. I mean, there are some efforts to do that, as you know, but it's similar to the problem of, okay, if you have a structure, how do you make an antibody to that specific epitope? It's a similar problem. But people, a lot of very smart people are working on this, and hopefully at some point we'll know more. Yes?
Really interested in your discussion of the N-terminus domain binding antibody, and I was wondering if you had any thoughts on why that was or neutralizing as opposed to other antibodies requiring to be RBD domain.
Well, there is a few NTD antibodies that are neutralizing, and I don't think anybody knows exactly why they're neutralizing, because nobody has really looked at the mechanism. Presumably, they're neutralizing because ultimately they affect the RBD domain and its ability, the conformational dynamics required to bind to the ACE2 receptor. But if you're asking for the precise molecular path by which this happens, what are the conformational changes, we don't know what those are. At least nobody has looked at them as far as I know.
I just saw that it was pretty close to the RBD, and maybe I thought it was a steric hindrance.
It's binding exactly. It affects the RBD dynamics, and the RBD domain can be up or down, and that makes a big difference. But how exactly? Nobody has looked at that, as far as I know.
I have
And
one
we
more
did.
question, which
Yes,
is,
that's
I
a
guess,
great
related
question.
to imprinting. Are there certain epitopes of proteins that B cells have a harder time making antibodies against, or easier time?
For sure, there are many epitopes for which it's very, very difficult to make antibodies. Exhibit A, HIV. The blocking antibodies, the neutralizing antibodies to HIV are super difficult to generate, to elicit, and that's why there are very few people there are a number of probably 10, 50% of HIV infected individuals that are elite neutralizers, but elite controllers that make large amounts of antibodies that can control the virus for a long time, they are really hard. That happens very rarely.
But that's not because of mutations? I thought that was difficult because of mutations, not because B cells generally have a harder time making
Well,
antibodies against a
not only
known
that, but
epitope.
they have to have a certain epitope. You have to bind a certain, any virus, any pathogen has to have some portions of its protein that are not mutated, because if these parts are mutated, then the virus can no longer survive. It can no longer perform its function. And those are the ones that you need to target. But some of these, because viruses are smart, and by the way, they evolve much faster than we do. Viruses are smart in some ways. There are many of these epitopes are not as easy to elicit an immune response. They require very gnarly antibodies with very long CDR sequences, loops that penetrate into a binding pocket in the antigen and so on. Another thing that blows my mind, and I didn't believe it because I don't believe a lot of things, and then say, oh, crap, should have believed it earlier, is the fact that we have, and I didn't show any data, a lot of antibodies. Like there are billions of different antibodies that we can make. But if you look at flu, for example, because we've done that, many of us share some antibodies that are exactly the same. And how the hell does that work? It shouldn't. The math say that it shouldn't, but it does. And these are called public antibodies. They've been seen at the B cell level, and we said, well, it's not really real. But now we're seeing them in the serum, and anyway, that's why I said there are a lot of things that we don't know about, especially circulating antibodies. The other thing that we don't know about circulating antibodies is why on earth how can we make vaccines that can prime antibodies that will protect us for life? You get vaccinated once, no booster, or maybe one booster, and then you prime for life, like the polio vaccine, or the smallpox vaccine, or the yellow fever vaccine. Well, there are some reasons, but in general, or even a hepatitis vaccine, which is subunit vaccine. Why does it work so well? And other vaccines, we're working on the norovirus vaccine. Norovirus is the really bad cruise ship diarrhea. I'll never get on a cruise ship just because I'm working with norovirus. For other reasons too. But anyway, all the experimental vaccines give you protection that lasts a few months. So they're not particularly good vaccines. I don't know why. So that's a different topic for what makes some vaccine response or antibody responses to home in and give you long-term serological protection, long-term production of antibodies.
Hi, thank you for the great talk. I have a question that's also more of a two-parter. First one is, for their vaccine research from COVID, or pre-vaccine research, how did acquired immunity play a role in the binding of just the antibodies to the spike proteins?
How did the what be?
How did acquired immunity play a role in your pre-vaccine data?
My acquired immunity?
Yeah, that's like when someone gets COVID.
Yes.
This was before the vaccine, and then they have the antibodies from getting the virus.
Or if they have, wait a minute, if they have infection and then immunize.
And then
the immunization, let me show the data, diverses the repertoire.
Diverses the repertoire, okay.
Diverses the repertoire. Infection restricts the repertoire and polarizes the repertoire to S2 antibodies. Actually, if you look at the publication literature carefully, you actually can see that has been even seen with serological data, with bulk serology, but not very clearly. But it's very clear when you look at the repertoire level analysis.
Okay, that might play into my second question, though. And that's, how do the antibodies differ from natural infection versus the vaccine? Yeah, I think you talked about the abundance of the different types of antibodies, but
Well, for one thing,
how does
the vaccine gives you much higher levels of antibodies. And if you've been infected and vaccinated, we have a few donors in whom about 10% of all the antibody circulation are anti-COVID antibodies, like really high levels. Likewise, if you're vaccinated and then infected, you also get a very large spike in antibodies. People call that, there is a name for that, it's called hybrid immunity, okay? We don't know exactly why infection and vaccination are so synergistic, but they are. I don't know if I answered your question.
Yes, yeah, so the abundance, yes, but also the diversity of epitopes.
The diversity of epitopes, yes. As you, when you get infected and then vaccinated, you diversify the epitopes quite a bit more towards more protective responses.
Okay, thank you.
Mm-hmm.
Excellent. So we are at time, and I think it's time to thank our speaker, George. Thank you so much. And thank you. I hope you have a wonderful weekend.